Hi, my name is Kirsten Dimmler, and I'm a PhD student at the University of Minnesota. Today, I'm going to tell you about the work we're doing in the Equine Genetics and Genomics Lab on developing a genetic test for recurrent exertional rhabdomyolysis in horses. So, what is recurrent exertional rhabdomyolysis, or RER? Well, rhabdo means striped, which refers to the appearance of skeletal muscle cells. Myo means muscle, and lysis is when a cell breaks open. Exertional refers to exercise, and recurrent means that it happens repeatedly. Together, this is the repeated breakdown of striped muscle cells following exercise. The symptoms of RER include muscle pain, cramping, extreme stiffness, sweating, and coffee-colored urine. The skeletal muscle of a horse consists of thousands of cells called myocytes. When an episode of RER occurs, these myocytes begin to die, causing their cell membranes to break down and the subsequent release of the cell's contents into the bloodstream. These contents include potassium, phosphates, and various proteins including myoglobin, which is an oxygen-carrying protein similar to the hemoglobin found in red blood cells. These chemicals and proteins make their way through the bloodstream to the kidneys, where they cause dangerous inflammation due to their toxic effects. The presence of myoglobin is what causes the characteristic coffee-colored urine we see with episodes of RER. At this point, horses need liters and liters of IV fluids to prevent potentially fatal kidney failure. This painful disease affects primarily two breeds of racehorses, the standardbred and the thoroughbred. RER affects 4 in 10 standardbreds and 1 in 10 thoroughbreds. Across the United States, there are approximately 1 million standardbreds and thoroughbreds. Of these, 163,000 are affected with RER. Not only that, but each affected horse is estimated to have 2 episodes of RER each year. This means that horses suffer a total of 327,000 potentially life-threatening episodes of RER every year. So what actually causes RER? Well, RER is a complex genetic disease. This means that environmental factors, such as the horse's diet, activity level, and stress level, interact with genetic factors in the form of mutations in a horse's DNA to cause disease. In this project, we focus mainly on the genetic aspects of RER. To study the genetics of RER, we identify both healthy horses that don't have RER, or controls, and sick horses, or cases. We collect biological samples, usually blood or hair, and then isolate and sequence the DNA. Using DNA sequencing, we can determine the unique genetic code of each individual and identify mutations, or places in the code that are different between horses. We can then work to determine which of these mutations are contributing to disease risk and by how much. Because the number of mutations identified can number in the millions, we need to use computers in the form of machine learning algorithms to help us decide which mutations contribute to disease risk and by how much. The algorithm looks at each mutation and each combination of mutations and compares how often they're found in healthy horses versus sick horses. It also considers certain attributes of the mutations like their potential effect on protein coding and biological function. This generates complex decision trees that help us estimate how much of an effect each mutation has on disease risk. Once we have estimated how much each mutation contributes to disease risk, and we have identified which horses have which mutations, we can essentially add up the estimates to calculate the probability, or likelihood, that an individual horse will develop RER in its lifetime. In this case, our horse on the left has a 65% chance of developing RER in its lifetime. Next, we can use a horse's estimated risk to classify it into different risk categories and make specific dietary or breeding recommendations. This information will allow veterinarians and horse owners to identify at-risk horses before clinical signs develop, provide early intervention and altered management practices, and allow horse owners to make informed breeding decisions to improve the health of future generations. And with that, I would like to thank the amazing team of researchers both within and outside of the University of Minnesota that have made this important research possible.